Usually when we talk about biological systems, there's this underlying expectation of straightforward mechanical logic.
Right, like a clear cause and effect.
Exactly. You can think of it a lot like a car. You press the gas pedal, the engine revs, and the car moves forward. It's clean, predictable, and linear.
Which is the ideal,
Yeah.
A very neat linear pathway from a signal to an action.
Right, but imagine if one day you sit in your car, you press down on that gas pedal, and instead of driving the car forward, the pedal suddenly detaches itself.
Yeah, it would be a serious problem.
A huge problem. And then it multiplies. And then those dozens of gas pedals start building a massive concrete parking lot just to lock themselves inside.
I mean, it sounds totally absurd, but that is actually the absolute definition of a biological anomaly. It's a complete, profound hijacking of normal cellular function.
Well, welcome to this deep dive. Today, we're exploring a really groundbreaking 2026 paper from Nature Cell Biology, alongside a few supporting sources.
Yeah, and our focus today is on a specific type of kidney cancer called renal cell carcinoma, or RCC.
Right. And the core of what we're looking at today is how these cancer cells manipulate the fundamental laws of physics inside themselves, basically to survive their own toxic overactivity.
And they accomplish this survival trick by creating these microscopic, highly organized storage vaults deep inside the cell's nucleus.
So our mission today is to completely unpack this surprising phenomenon. We are going to explore every mechanism, the physical forces at play, and every tiny detail of what researchers are calling TEAD1 condensates.
We really want to figure out how cancer builds these vaults, what they put inside them, and why they even bother in the first place.
Okay, let's unpack this.
So to understand why this parking lot behavior is so incredibly strange, we first need to understand what that gas pedal normally does.
Right, the baseline function.
Exactly. The gas pedal in this scenario is a protein called TEAD1. In a healthy functioning cell, TEAD1 is the ultimate growth trigger.
And it sits at the end of a famous pathway, right? The Hippo pathway.
It does. It sits at the very end of the Hippo cellular signaling network.
Conceptually, that's basically the cell's braking system for growth, isn't it? Like, it stops things from getting out of control.
Yes, and it does that through a highly regulated chain of command. Normally, the Hippo pathway acts as a suppressor.
So it keeps the brakes firmly on.
Right. Without getting bogged down in the alphabet soup of specific enzymes, you can think of it like this. The cell tags its growth-promoting proteins with a specific chemical signal.
Okay.
And that chemical tag acts like a bouncer at a club, effectively locking those growth proteins out of the nucleus.
Oh, I see. They're forced to stay out in the cell's main body, the cytoplasm, completely inactive.
Exactly. So the brakes are on, and the cell doesn't unnecessarily divide.
But in renal cell carcinoma, this kidney cancer we're talking about, those brakes fail completely.
They really do, yeah. In this specific cancer, the structural scaffold of the braking system frequently mutates and falls apart. Things like NF2 or SAV1 mutations.
And because the brakes are broken, those growth proteins never get tagged by the bouncer.
Right, so they flood straight into the nucleus, and once inside, they bind directly to our gas pedal, TEAD1.
And they just start rapidly turning on genes that promote cell proliferation. They just slam on the gas.
They do. And that's what researchers saw when they started looking at patient-derived RCC cancer cells.
They looked at normal kidney cells, like HK2 cells, and then they looked at these highly mutated cancer cells, like UOK121.
And unsurprisingly, the levels of TEAD1 and its partner growth proteins in the cancer cells were absolutely through the roof.
But here is where the sources note something completely bizarre under the microscope.
Yeah, when they used advanced imaging on these cancer cells, they didn't just see a diffuse, even glow of TEAD1 floating around the nucleus doing its job.
Like a normal active workspace. I think they call those STCs, or small TEAD1 condensates.
Right, normally you just see those tiny STCs. But here they observed TEAD1 physically clumping together to form these massive micrometre-sized droplets.
The massive ones. The sources call them LTCs, large TEAD1 condensates. The cancer cells had up to 5 of these massive droplets per nucleus.
Which is a huge structural anomaly.
But, seeing a droplet under a microscope is one thing. Figuring out what it's actually made of is another.
Definitely.
If I see a clump in a cell, my first instinct is that it's a solid aggregate, like a microscopic hairball of junk protein.
Which happens a lot in disease states like Alzheimer's.
Right. So how did the researchers prove it was a dynamic liquid droplet instead of a solid rock?
Well, they used a brilliant technique called FRA, which stands for fluorescence recovery after photobleaching.
I love the concept of FRP. It's almost like taking an incredibly powerful flashlight, blinding a specific tiny spot of a glowing droplet, and then watching to see if the glow flows back into the dark spot.
That is exactly how it works. It's essentially a way to test the physical state of matter inside a living cell.
Because if the structure is a solid rock, the spot you hit with the laser stays dark forever.
Right, the molecules are frozen in place. But here, they did a half-bleach FRAP. They blasted only half of the TEAD1 droplet with the laser.
And what happened?
Almost instantly, the fluorescence recovered and filled the dark space. That proved the TEAD1 molecules inside were actively swirling, swimming, and mixing with each other like a liquid.
That's wild. And didn't they bleach the whole droplet too?
They did. They did a full bleach FRIP, blasting the entire droplet so it was completely dark.
And it recovered.
It recovered its glow with a half-life of just over 3 seconds.
Wait, 3 seconds? That's so fast.
Right, which tells us TEAD1 is constantly, rapidly exchanging itself with the fluid outside the droplet. It's highly dynamic. It breathes.
So we definitely have a liquid droplet. But wait, usually when we talk about liquid droplets forming spontaneously in a watery environment like a cell, we are talking about hydrophobic interactions.
The classic oil and water separation.
Exactly, like the fat droplets floating in chicken soup. If these are just oil-like droplets, couldn't the researchers just dissolve them? Did they try washing them with a cellular soap?
That was their exact next thought. In the lab, there is a specific chemical called 1,6-hexanedyl.
Which is basically dish soap for a cell, right?
Pretty much. It specifically breaks up those weak hydrophobic oil and water bonds. So they applied a heavy dose of it directly to the living cells.
And the droplets melted?
No, they didn't shrink.
Yeah.
They didn't dissolve at all.
Oh, wow.
Yeah, their numbers didn't drop. They were completely unaffected by it.
So if a chemical designed to break liquid droplets completely fails, that means a totally different non-hydrophobic physics mechanism is holding these things together.
Exactly. It's a massive clue that we aren't just looking at random cellular oil spots.
Okay, let's do a quick bullet recap. TEAD1 is normally a growth trigger, but in this cancer, it forms giant liquid droplets, these LTCs. And these droplets aren't simple hydrophobic oil spots.
That's the baseline of the mystery.
Which leaves a huge question for you, the listener, to think about. If you were a cancer cell, why would you take your primary growth-driving protein and lock it up in a giant droplet? What are you trying to hide from?
To answer that, to figure out what this unbreakable droplet is actually doing, we really need to look at where it's hiding.
Right, location is everything. TEAD1 is a transcription factor. Its entire identity, its only job, is to bind to DNA and print out RNA to make the cell grow.
That's its sole purpose.
So logically, these massive droplets must be incredibly busy factories, right?
That is exactly what you would assume. But the most shocking discovery in this paper was what these droplets were not doing.
What weren't they doing?
They were utterly, completely dead zones.
Wait, really? A massive cluster of growth proteins, and they aren't turning on any genes. How do they prove that?
By looking for the physical machinery of transcription. Normally, when a gene is turned on, a massive molecular machine called RNA polymerase II clamps onto the DNA.
And physically prints the new RNA strand.
Right. Specifically, the serine 2 phosphorylated version. The researchers stained the cells specifically looking for the active version of that machine.
And it was there.
It was completely absent for the droplets. They also looked for the chemical tags that mark active DNA enhancer regions like H3K27C. Absent. BRD4. Absent.
So the printing presses simply aren't there.
To be absolutely certain, they fed the cells a chemical tag called 5-ethynyluridine. It gets physically built into brand new nascent RNA as it's being made.
So if the droplet was making RNA, it would glow with this tag.
Exactly. And the droplets were completely dark. They excluded all markers of active transcription.
That's crazy.
They didn't even overlap with the classic canonical target genes that TEAD1 is famous for turning on, like CYR61.
So it isn't an active workspace. It's not a factory. It's an attic. It's just a dead zone.
A very deliberate dead zone.
But where exactly in the massive landscape of the nucleus is this attic located?
The imaging showed they sit right next to the nucleolus, or the nuclear lamina. But to get really specific, the researchers stained the DNA for epigenetic markers.
Okay, looking at the actual packing of the DNA.
Right, and what's fascinating here is they found the droplets perfectly colocalized with tightly packed, highly condensed, and totally silent DNA.
Markers like H3K99E3, right?
Yes, exactly. H3K93 is a classic marker for heterochromatin.
So it's actively seeking out the quietest parts of the genome.
Yes, and its physical address is fascinating. Imagine a single chromosome. We often picture it as an X shape.
Right, the classic X.
The pinched waist right in the middle of that X is called the centromere. The densely packed, highly structural, silent DNA sitting immediately adjacent to that waist is called the pericentromeric heterochromatin.
And that's marked by CENPA, right? That sounds like the ultimate hard-to-reach genomic attic.
It really is. It's packed tight and usually closed for business. It is highly structural and normally completely devoid of transcription factors.
Yet the researchers found that TEAD1 is parking itself exactly 0.2 micrometers away from that specific centromere waist.
It actively chooses this pericentromeric attic, ignoring the ends of the chromosomes entirely.
But TEAD1 isn't the only protein that forms little liquid bodies in the nucleus. Did the researchers check if TEAD1 was just crashing someone else's party?
Oh, you mean like overlapping with other structures?
Yeah, do these droplets overlap with other known nuclear structures, like
Yeah.
Cajal bodies, which process RNA?
They checked all the usual suspects. Cajal bodies, nuclear speckles, polycomb bodies, which also silence DNA. The answer was definitively no across the board. The TEAD1 condensate doesn't overlap with any of them.
So this is a completely unique, specialized, standalone structure.
Entirely unique.
Okay, let's step back and do another recap. These LTCs are transcriptionally dead. They avoid active genes, and they park themselves in the dense, quiet attic of the pericentromeric heterochromatin.
That sums it up perfectly.
Which brings up a great question for you to think about. Think about how hard it must be to access a tightly packed genomic attic. How does a protein like TEAD1 even know exactly where to anchor itself in that dense jungle of DNA?
And the mechanics of that anchoring process is where the paper gets really elegant.
Because we know where they form, but we need to explain how they anchor there.
Right. The researchers hypothesized that even though TEAD1 isn't turning on genes, it must still be physically grabbing the DNA to start building this droplet.
Because TEAD1 has a very specific shape, right? A highly conserved DNA binding domain made of coiled alpha helices.
Exactly. So they needed to test if the physical grip is the key.
How do you test that? Do you just break its fingers?
Essentially, yes. They engineered mutant versions of the TEAD1 protein, specifically messing with that DNA binding domain.
Like the delta 55 to 122 mutant or the H1 and H3 mutants.
Yes, in those variants, they took the section of the protein that normally grips the DNA, and they swapped out a few normal amino acids for an amino acid called proline.
Why proline specifically?
Well, in biochemistry, proline is famous as a helix breaker.
Because of its shape.
Right, because of its rigid ring structure, it physically kinks and bends the protein out of shape. By swapping it in, they warped the gripping mechanism.
So the protein could no longer physically lock onto the DNA.
Exactly. They completely broke the grip. They also tested mutating other parts, like the YAP binding site, but that didn't matter.
And what happened to our giant liquid droplets when the DNA gripping fingers were broken?
They vanished. The massive condensates completely stopped forming, and the protein stopped associating with that H3K9-AME3 heterochromatin.
Wow. So physical binding to the DNA is absolutely required to build the structure.
It's the mandatory first step.
But it can't just be any DNA. It's landing in a very specific zip code. What exact sequence of DNA letters is it looking for?
Well, TEAD1 naturally searches the vast human genome for a very specific short sequence of letters called the MCAT motif.
And they found this by unpacking the QP-seq and RNA-seq data. Right?
Yes. When they mapped out exactly where TEAD1 was attaching, they combined it with computational analysis of the genome sequence. And they found something wild.
What did they find?
That pericentromeric attic region isn't just random structural junk DNA. It contains a massive, dense, repetitive clustering of these MCAT motifs.
And just as a control, they checked other motifs, like AP1, and those weren't clustered there.
Right. It was specific to the MCAT motifs.
Here's where it gets really, really interesting. Think of the physics of what's happening. It's like TEAD1 is a tennis ball, and it's covered in very, very weak Velcro.
Okay, I like this analogy.
When these balls are just floating around the nucleus, they bump into each other, but the Velcro is too weak to hold them together. They just bounce off.
Right, they don't stick to themselves much.
But that specific attic DNA, that pericentromeric region, is a giant solid wall of matching Velcro. That's the clustered MCAT motifs.
And once a few TEAD1 molecules hit that wall and stick to it, the local density completely changes.
Right. Now you have a bunch of these weak Velcro balls crowded tightly together in one spot. Suddenly, their weak interactions with each other are enough to take over.
They form a massive crowd, The DNA acts as a structural scaffold, triggering that massive crowd to form out of thin air.
It's beautiful.
It really is. That is the exact mechanism of liquid phase separation in this context. The high local concentration on the DNA scaffold pushes the physics of the system past a tipping point.
And the liquid droplet condenses out of the surrounding fluid.
The researchers even proved this with an in vitro validation. They mixed purified TEAD1 with synthetic DNA in a test tube.
And what happened?
Well, DNA with 0 or just one MCAT motif did almost nothing. Barely any droplets formed. But synthetic DNA engineered with a tight cluster of 4 MSAT motifs, the 4X MCAT.
The concentrated Velcro?
Yes. It caused a massive explosion of condensate formation. The clustered scaffold is the ultimate trigger.
So to quickly recap, condensate formation requires TEAD1's DNA binding domain. The pericentromeric DNA is packed with MSAT motifs, and these clustered motifs act as a scaffold to trigger phase separation.
Exactly.
Which leaves us with the ultimate question for you. Now that we know how they are built, we have to ask the ultimate why. Nature doesn't build complex structures by accident. What advantage does a cancer cell gain by hoarding TEAD1?
And that brings us to the depot theory. It all comes back to the broken breaks we discussed at the very beginning.
Right, the Hippo pathway mutation.
Renal cell carcinoma has a severe, potentially fatal problem. The mutation means the cell is constantly flooded with TEAD1 and YAP. And in biology, too much of a good thing is highly toxic.
Because the cell can't just grow infinitely without consequence. What happens if TEAD1 actually does its job too well?
If TEAD1 is allowed to run wild and activate its target genes at maximum capacity, the cell's internal machinery goes into absolute overdrive. This massive metabolic stress triggers something called ferroptosis.
Ferroptosis, that's an iron-dependent form of programmed cell death, isn't it?
It is. The cell literally burns itself out and poisons itself.
So the hyperactivity will quite literally kill the cancer cell? The cancer cell is just trying to save itself from burning out?
Yes. By shoving all that dangerous excess TEAD1 into these silent droplets, the cancer cell is buffering its own system.
It's building a microscopic panic room, or really it's more like a high-security offshore bank account.
I like the bank account analogy.
Yeah, the cancer cell has too much dangerous inventory, so it locks it away where it can't be spent, ensuring the local cellular economy doesn't undergo hyperinflation and crash the whole system.
That is a perfect visualization. And the researchers actually proved it acts as a quarantine zone, using single particle tracking.
Which is where they use highly advanced microscopes to watch individual single TEAD1 molecules moving in real time.
Right. They observed that when a molecule wanders into one of these large condensates, its residence time, basically how long it stays before leaving, is significantly longer than when it's just floating in the nucleoplasm.
It goes in and it gets stuck.
Exactly. It's trapped inside the LTCs.
But a bank account is only useful if you can prove the money is actually in there. Did they find a way to pop the vault and flood the economy?
They did a really brilliant stress test to prove it acts as a buffer. They exposed the cancer cells to hyperosmotic stress.
By adding specific salts or sugars like sorbitol or sodium chloride outside the cell.
Right, which rapidly draws water out of the nucleus.
And that changes the physical crowding inside the cell, altering the physical forces that hold the droplet together.
Exactly. This sudden change dissolved the LTCs entirely. The vaults melted, and all that trapped TEAD1 was instantly released back into the main fluid.
And what happened to the cell's gene expression when the vault popped?
They saw a massive immediate spike in the expression of the target genes, specifically CYR61.
Wow. The buffer was gone and the system was flooded.
The depot popped and the genes went completely wild.
That is incredible. The droplet is literally the only thing keeping the cancer cell alive from its own mutations.
It's a pure survival mechanism.
Let's do a quick bullet recap. Condensates act as depots to buffer excess TEAD1. Trapping the protein prevents lethal overactivation of genes that cause ferroptosis. And breaking the depots floods the cell and spikes gene activity.
That covers the why.
So here's a question for you as a listener. If you were designing a cancer drug, would you want to stabilize these depots to keep the cancer quiet or blow them up to force the cancer cell into ferroptosis?
Before you answer that, though, we have to look at how sophisticated this vault actually is.
Right. Is it just a mindless trash bag for excess protein? Is this depot just a junk drawer? Or is it a selective, highly curated VIP room?
To figure that out, the researchers had to see what else gets to go inside, which is incredibly difficult, looking inside a microscopic liquid droplet.
How did they manage it?
They used a cutting-edge proximity labeling technique, utilizing a tool called Mini Turbo.
Walk us through how mini-turbo actually works, because it sounds like science fiction.
It's a genetic engineering marvel. They took a biotin ligase enzyme called mini-turbo, which is basically a chemical tagger,
Okay.
and they physically attached it to the TEAD1 protein. Then they feed the live cells a chemical called biotin.
For how long?
Just 10 short minutes. During that time, the mini-turbo enzyme wakes up and aggressively starts slapping biotin tags onto any protein sitting immediately next to TEAD1 inside the condensate.
I picture it like giving TEAD1 a paintball gun. For 10 minutes, it just spins around and shoots biotin paint at absolutely anyone standing close enough to it inside the VIP room.
Yes, and once the 10 minutes are up, they use mass spectrometry to pull out all the tagged proteins to see exactly who was inside the vault.
But wait, how do they know the proteins they caught were actually inside the droplet and not just random proteins TEAD1 bumped into while walking down the hallway?
That requires a clever control. They compared the proteins painted by the wild-type droplet-forming TEAD1 against the proteins painted by the H3 mutant TEAD1.
Oh, the kinked one that we know cannot form the droplets.
Exactly. Anything painted by both was just hallway traffic. Anything painted only by the wild-type TEAD1 was an exclusive resident of the vault.
And what were the findings? What did the guest list look like?
They found 186 proteins specifically and exclusively enriched inside the functional condensates.
186.
And if we connect this to the bigger picture, these weren't random. They found selective protein incorporation.
So it's actively recruiting a specific staff. One of the major ones the paper highlights is a protein called RPA2, right?
Yes. RPA2 is a crucial component of a complex that maintains genome stability.
So the researchers naturally asked if RPA2 was acting like a brick holding the vault together, or if it was just a visiting worker.
Right. Is it a scaffold or a client? They tested this by knocking down RPA2 entirely.
Yeah.
The TEAD1 condensates still formed perfectly fine without it.
So RPA2 isn't a structural scaffold. It's a client. It's a guest.
Furthermore, when they looked at the movement speeds, RPA2 moves in and out of the droplet much faster than TEAD1 does. It's visiting the vault, doing a job, and leaving.
So it is absolutely not a spam folder or a junk drawer. It is a highly curated VIP lounge.
Highly organized.
Let's recap this section. They used mini-turbo to map the condensate's neighbors. They found it selectively imports specific proteins. And RPA2 lives inside, likely to help maintain the structural integrity of that tightly packed DNA.
It's bringing in specific maintenance workers.
Which makes you wonder, how many other transcription factors might have secret moonlighting jobs, forming storage droplets and recruiting totally different proteins than they do when they are transcribing genes?
That's a huge question.
So bringing this all together, what does this all mean for us?
It changes everything about how we view the biology of a cancer cell. TEAD1 is no longer just a one-dimensional gas pedal. It's a structural architect.
It creates its own storage vaults in the pericentromeric attic to protect cancer cells from their own hyperactivity.
And it completely flips how we view inactive regions of DNA. They aren't just dead space or junk. They are vital, highly organized storage racks.
And the therapeutic implications here are immense, aren't they?
Massive.
Yeah.
Right now, condensate targeting therapies are a huge new frontier in medicine. Drug companies are scrambling to design chemicals that can dissolve these rogue liquid droplets.
But this paper is a massive warning sign. We must be incredibly careful.
Because if a drug accidentally bursts these specific TEAD1 depots in a patient with kidney cancer, they aren't destroying the cancer. They might flood the cell with growth signals and cause the tumor to explode.
Or, conversely, if we understand it perfectly, we might intentionally pop it to push the cell into fatal ferroptosis burnout. We have to know exactly what the droplet does before we try to pop it.
It's a microscopic tightrope walk.
It really is. Which brings us to a final thought for you to mull over on your own. We've spent this entire time talking about how a broken cancer cell hijacks the laws of phase separation to build microscopic panagrams for its dangerous proteins.
Right.
But nature is highly efficient. If a cancer cell can do this, what are your perfectly healthy cells using these liquid droplets for right now that we haven't even discovered yet? Are there thousands of distinct, invisible liquid micro-worlds winking in and out of existence inside you every single second?
Something to think about.
Definitely something to think about the next time you sit down and start your car.